All right, you've had your students pick up their plates from the previous week. You have them lay them out on the desk. You pass out the UV light, or you walk around with it, and they scan these and they see that this is very much reactive to UV. This LB amp era plate, very much reactive to UV. And so then you say, all right, we're going to analyze what we did in essentially 3 experiments. Experiment 1 is going to involve these 2 plates. Experiment 2, these 2 plates. Experiment 3, these 2 plates. So how are you going to do that? Well, here we go. Again, all the lids are on, but you have the students take them off one at a time and examine them for bacterial growth. When they take off the lid of this one, what are they going to find? Well, someone may say there's no bacteria growing. The problem is it's covered completely smoothly with bacteria, and they don't recognize that. So you may have to help them out, but there should definitely be lots and lots of bacteria, maybe the whole thing completely covered with bacteria growing on this one. So we put that lid back on. We have them check this one. Any bacteria growing on that one? Absolutely not. And so, what's the difference maker? Well, I guess it's this AMP stuff. What is AMP? Well, that's ampicillin, and it was mixed in with a growth gel. And so, we need to draw a conclusion about the bacteria. Now, there's some bacteria that came off the starter plate that went in both of these, so it's the same bacteria all the way through. But what can we say about these bacteria based on this comparison right here? The bacteria what? And I'd ask students, what can you come up with? And they'll probably say something about ampicillin and so forth, and you may have to help a little bit. But the conclusion, I would think, would be what? I would say that these bacteria that were in these things are not ampicillin resistant. Is that right? They grow fine when there's no ampicillin. They don't grow at all when there is ampicillin. So again, the conclusion from experiment number 1, you could call it, is the bacteria are not ampicillin resistant. All right, now we move over to this set right here. And we've already checked this one out, so now we check this one. And students take off the lid. Oh, by golly, lots of bacteria growing on that one. The same bacteria that was put in both these, is now growing. Well, what's the difference maker? The difference maker, I guess, is that plus sign. What does that plus sign mean again? What was put in the plus capsule that wasn't put in the minus capsule? Well, P-glow. P-glow was put in it. And so P-glow is what? It's a plasmid, a little circle of DNA. And so what conclusion can we then draw about P-glow? Well, the students, they're not thinking too hard about this. They haven't reviewed their notes from the previous week probably. So they may say P-glow has ampicillin resistance. What? All by itself? It's ampicillin resistant? No. It's just a plasmid. It's not alive. It's just a little DNA molecule. P-glow can't be ampicillin resistant. It's just a little plasmid. But what did we try to do? What was the whole purpose of this experiment? The whole purpose of the experiment was to get P-glow inside what? Inside bacteria cells. And so P-glow, if everything worked right, we had bacteria cells, this representing a cell, and P-glow being a plasmid, we attempted to get P-glow inside, and apparently when we did, what happened? The bacteria can live in the presence of ampicillin. So what can we conclude about P-glow then? P-glow must have some way of providing ampicillin resistance, and based on what we've studied before, what can we say? We can say that P-glow must have what kind of gene in it? Yeah, an AMP resistance gene. Ampicillin resistance gene. So P-glow has an ampicillin resistance gene. Now you might ask them to look at that plate a little bit. Said, is that covered over like this first one? No, there's probably going to be little circles. Each one a colony that grew from one bacteria cell that picked up the plasmid. And so thousands of bacteria died, millions maybe of bacteria died on this plate that didn't get P-glow. The only ones that survived and grew a little colony were the ones that did what? That picked up the P-glow plasmid. So there you have it. So again, comparing these two, what can we say about piglow? The piglow plasmid provides what? Ampicillin resistance, and that must be because, based on what we've learned earlier, it has what? An ampicillin resistance gene in that plasmid. Alright, so that's those two. Now we come to the last two. And obviously there are bacteria growing here, and they're somehow there's some reaction to UV. They're a beautiful little translucent green there. What's the difference maker here? Well, it's arabinose. What is arabinose? It's a sugar. What would be a simplistic conclusion then? Arabinose reacts to UV light? That would be a great conclusion, except it would be totally wrong. And so now we need to kind of rely on something we learned in the material that came with the lab. And that is, piglow not only has an ampicillin resistance gene, piglow has a gene that came from what organism? A jellyfish. And what gene is that? It came from the jellyfish. It is one that is called the GFP gene. GFP gene. And the GFP gene, what does GFP stand for? It stands for green fluorescence protein. And so this is a gene that when protein synthesis takes place, it produces green fluorescence protein. And that is what reacts to the UV, the GFP protein that is produced by the GFP gene and so forth through protein synthesis. Now the question is, there's no green fluorescence protein in this one, but did the bacteria that lived over here, did they get the GFP gene? And students say, nah. Well, wait a minute now. Wait a minute. They got piglow, right, or they wouldn't be alive. Piglow has an ampicillin resistance gene, but isn't the GFP gene along for the ride as well on this piglow plasmid? Did these guys get the GFP gene? Yes, they did, but it's not working. And now we can probably figure out what the role of arabinose is in this experiment. What is the role of arabinose? It was in your handout last week. It's probably been a while since you read it. But what is arabinose? What's the role of arabinose with respect to this GFP gene? Without arabinose, the GFP gene just sits here and does nothing. With arabinose, the GFP gene kicks into gear, and you get all this GFP. It looks real pretty, glows in the dark. So what can we say about arabinose and the GFP gene? And usually a student will answer. They use various words. The words I like, and sometimes a student will come up with this, the arabinose activates the GFP gene. And so without arabinose, the GFP gene sits there, silent. With arabinose, the GFP gene kicks into gear and produces GFP, and you get this pretty color. Alright, so what conclusions did we draw? First conclusion, this bacteria that was used throughout this experiment by itself is what? Ampicillin resistant or not? Not ampicillin resistant. Comparing these two, you learn something about piglow. What did we learn about piglow? It provides ampicillin resistance because why? Because, wow, it must have an ampicillin resistance gene, right? And then we compared these last two and saw arabinose, and we saw UV reaction over here. And so what does the arabinose have to do with the UV reaction? Well, we see it has something to do with the GFP gene. We had to read about that. But it has something to do with the GFP gene. The GFP gene is part of piglow, so these bacteria that lived here got the GFP gene. These bacteria here got the GFP gene. Here the GFP gene was silent. Here the GFP gene was active. So what kicked the GFP gene into gear? What activated it must be that arabinose. And that, ladies and gentlemen, are all the things we can conclude from this little experiment. All right. Bye-bye.